So in today's video, we're going to be talking about cell communication. And first, we're going to be talking about local regulators and hormones. So local regulators signal nearby adjacent cells, so their message is received by nearby cells and their short distance. Hormones, on the other hand, are chemicals released in a part of the body and travels through the bloodstream and affects other parts in the body. So this can be thought as of like the long distance signaling. Next, we're going to be talking about hormones, and the two hormones I have here are a protein-based hormone and a lipid-based hormone. And the differences are that a protein-based hormone is made up of chains of amino acids, or a polypeptide hormone. And examples of this are ADH, which is the antidiuretic hormone. And for a lipid-based hormone, they're derived from lipids, as in the name, and they're mostly known as steroid hormones. So examples of this include testosterone or estrogen. Cell communication can be simplified into 3 steps. So the first one being cell reception, then transduction, and then finally the cell response. There are 3 different types of protein receptors. The first one being the G protein link receptors, the receptor tyrosine kinase, and the ion channel receptor. So we're going to go into more depth about the G protein link receptor, and the parts are the ligand, the G protein link receptor, the G protein with an attached GDP, and the enzyme. So the first step of this receptor is that when the ligand attaches to the receptor right here, it'll attach into this site right here. It goes under a conformational change, which means it changes in shape, which means, so basically when it changes in shape, it's like hugging the ligand, so it has more like a tighter fit and they link up together perfectly. And the inactive, so how we know when it's inactive is that it will show up as a GDP, but then once the ligand attaches, this G protein, the inactive G protein, will come over here and it will diffuse across the cell membrane to the G protein link receptor with the ligand attached, and it's going to activate it. And when it's activated, it exchanges GDP into GTP. So once the G protein moves over here, you will no longer see the GDP, but it will be replaced with GTP, and that's how you know it's active. Once we're still over here with the GDP and the G protein, the ligand will actually detach, and the receptor, the G protein link receptor, will return to its original shape, and then the active G protein with the GTP will come over here, diffuse across the cell membrane to the enzyme and activate it. Once the G protein activates this enzyme, it will undergo another conformational change, and then this will lead to the cell response. Not directly, but it'll create something. So for example, the cell response from the enzyme will be to make cyclic AMP, and that will be used to activate the transduction pathway. And now you're thinking, how, is this always going to be active? How can it return to its original state? And it returns to its original inactive state. So the active G protein will hydrolyze, so it would break off from the enzyme, and once it hydrolyzes and breaks off, it will turn back to its inactive form, and it would turn back to GDP instead of GTP. So the next protein receptor is the receptor tyrosine kinase. So you're probably thinking this is way different from the G protein linked receptor. So these are actually polypeptides, these two, and then they also have tyrosine regions attached to them. And these are actually, and the tyrosine kinase will actually transfer phosphate groups. So the definition of a kinase is an enzyme that catalyzes the transfers of phosphate groups. So this will actually do that. And this is the binding site, and this is the ligand that will attach to it to carry out the message. So the first step is that when the signaling molecule attaches to the binding site, these two will actually come together. So they'll be right next to each other, they'll be adjacent to each other, and they'll form what's called a dimer. They will also undergo a conformational change, and the dimer is actually unphosphorylated because there is no phosphate group attached to these tyrosine regions, but the tyrosine regions are activated. In receptor tyrosine kinase, it'll actually utilize ATP, so it'll turn ATP into ADP for a more visual. So this will be ATP, and then it'll turn into ADP plus a phosphate, because these together will create ATP. Then that phosphate group will attach to each of these tyrosine regions right here. So there will be 6 of them in total. So just for visual, we'll have phosphate groups attached to each of them. And same on this side. So the phosphate groups are attached. After the phosphate groups are attached, inactive relay proteins will come in to each and every single one of these. So let's say that's a relay protein. It'll come to each and every one of these. So it would surround it, and they're inactive, but once they attach to the phosphate group, they'll undergo a conformational change and they'll become active. The cool thing about the tyrosine kinase relay proteins is that once they attach to the phosphate groups, they actually don't, well, they could, but they actually could vary up to different cell responses. So once this attaches, this could have a different cellular response than these two. So you can have up to 10 different responses for one single binding event. And last but not least is ion channel receptors, and these are fairly easy. Okay, so we're going to have this ligand right here, and then the gate, this is like an ion gate, so it's closed, so the ions cannot flow from their concentration gradient into the cell. So the ligand's job is so when it attaches right here, it will not go like inside, it will just attach to the ion gate, and once it attaches, it undergoes a conformational change. So that means once it attaches, this gate, you can think of it as a gate, it'll actually open. So this will flip open, and then now the ions can flow in from their concentration gradient into the cell. And the cell response is when the ions flow through, and once the ion detaches, it'll close. So this is kind of like a lock and key. So we're going to return to the G protein link receptors, and we're going to talk about secondary messengers and their job. So a G protein link receptor, an example of this cellular response could be, here, let's write it down here. The cellular response from the first messenger could be to create cyclic AMP. So, and cyclic AMP is produced from the first messenger. So cyclic AMP's job is actually, it's going to be the second messenger in the G protein link receptor, and then the second messenger will actually activate the transduction pathway, the signaling transduction pathway. Okay, since we're done with that, we're going to move on to the transduction pathway, which is step number 2 in cell communication. So as we talked about in the G protein link receptor, the first messenger was to create cyclic AMP. This is just an example. And transduction is the same as in all 3 protein receptors. So this part, so the signaling transduction phosphate is the same in all 3, but the message is obviously different for each of the signalings. So for example, we're going to use cyclic AMP. So cyclic AMP is a second messenger, and this will activate the phosphate cascade. This is called the phosphate cascade. And then right here it says the first messenger response initiates the phosphorylation cascade. So that would have been the cyclic AMP. So that will actually activate this right here. This is protein kinase A. This is protein kinase 2, protein kinase 3. And remember, kinase, it activates the transfers of phosphate groups. So we're going to imagine cyclic AMP over here, and then once it attaches to pKa, it'll actually become active. And then protein kinase A hydrolyzes ATP into ADP and attaches the phosphate group to pK2. So in this ligand, the tyrosine kinase receptors, it's going to utilize ATP. So it's going to break down ATP into ADP and then that phosphate group, and then it's going to attach that phosphate group and keep on going down. So once that phosphate group attaches, it undergoes a conformational change in shape, and it's now active. Once it's active, it can now repeat the same step. So we're going to take ATP, break it down into ADP and that phosphate group, and then it's going to attach to pK3 and undergo another conformational change. And for the last time, we're going to take another ATP, break it down to ADP and that phosphate group, but this time there's no more kinases. This time that phosphate group is going to activate. This was once inactive protein, but once that phosphate group attaches, it's now an active protein. That active protein will result in a cellular response for whatever the cells are trying to communicate, and that's the end of cell communication and transduction. And you're probably thinking to yourself, are all these phosphates going to stay attached and are active? Well, the answer to that is that an enzyme, protein phosphatates, detaches the phosphate groups off of pK2 and pK3, so they return to their inactive form, so they can repeat this over and over again. So there's actually many different types of cellular responses, but we're going to talk about 3. So an example of a cellular response from a G protein link receptor could be epinephrine. This is not avoid the cyclic AMP, but epinephrine is also known as adrenaline. So this will be utilized in the fight or flight response that the body gets. So the ligand for epinephrine will attach, and their cellular response after they go through the first messenger and then also the transduction pathway, their cellular response will be to release sugar into the bloodstream, so that can utilize to make more ATP to run away from harm or just alert the body. Another example could be a growth factor. So let's say in blood clotting, the blood clotting messenger will link up to the G protein link receptor, and its cellular response after transduction will be to produce platelets to actually close that wound and form a clot, or a scab, I mean. Another example could be a protein-based hormone, and since it's a protein, it cannot diffuse through the membrane easily, so it'll attach to a surface protein receptor, and then its cellular response would be to, for example, it would produce ADH or antidiuretic hormone, and that would, so it would produce that when the body has a high osmolarity, so it'll actually produce that to retain all the water and not lose any until that person has drinking more water and the osmolarity goes down. Thanks for watching!